Hello everyone, welcome to NA's Institute, a complete chemistry classroom program. This is the third class of bioinorganic chemistry. In this class, we'll mainly consider the sodium-potassium pump. We will learn what is sodium-potassium pump, how does it function, what do you mean by evolution in the activity of this sodium-potassium pump, how does vanadate interfere with the activity of the sodium-potassium pump, and selectivity of the sodium-potassium pump in transporting the sodium plus and potassium plus ions. Okay, so prior to discuss about the sodium potassium pump, I would like to inform you that for most of the animal cells, the concentration of this potassium ion inside the cell is higher, whereas the concentration of sodium plus ion outside the cell is higher. This is okay. But how much then should be the concentration of the sodium and potassium ion with respect to the inside of the cell and the outside of the cell? Now, for a typical cell, the concentration of sodium ion outside with respect to the inside of the cell is 15, whereas concentration of K plus ion inside the cell with respect to the outside of the cell is almost 25. So it is just the opposite case. So the concentration of the sodium ion inside the cell is comparatively very less, whereas the concentration of this potassium ion inside the cell is comparatively very large. But why such a condition exists? Because the high concentration of this potassium ion inside the cell is required for some vital process occurring within the cells. Say for glucose metabolism, say for protein biosynthesis, many kind of bioinorganic reactions takes place just because of this ratio. So we are dependent on this kind of ratio. Because of this ratio, different type of reactions, different type of bioinorganic process are going on inside our system, inside the human bodies also. Now the next question comes that how to maintain this kind of concentration. Because see, as the concentration is very much vital, then we have to maintain the concentration. Otherwise, the vital process, the vital reactions would result to end at that position. So we will have to maintain that kind of concentration. Now the importance of this sodium potassium pump comes into the picture. Now, whenever we are just hearing these term or just pointing out this term about sodium potassium pump, so definitely we should think that there should be one kind of driving force which is running this kind of sodium potassium pump, right? So it is driven by the internal enzyme. Internal enzyme means that the enzyme is already present inside the biological system, or you can say inside the body, inside the human body, and that enzyme is called or named as sodium potassium ATPase. Actually, the energy is obtained from this hydrolysis of ADP, that is ATP is converted to ADP to run the active transport process. Here I have shown you a typical diagram of a sodium potassium ATPase, that is the internal enzyme. There are two alpha units and there are two beta units also. And here you can see that there is one extracellular pod and there is one side is the extracellular pod and another side is the inside of that cell. So you can see that inside of the cell, the concentration of sodium is 30 millimoles, whereas outside of the cell concentration of sodium is 150 millimoles. So you can have a question that in these case actually the concentration of sodium outside versus inside would be just 5, whereas in case of potassium the concentration of these inside versus outside would be 30. But we have just learned in the last slide that the concentration is some kind of different. That is, a concentration of sodium outside versus inside was around 15, whereas in case of potassium inside versus outside was around 25. So I have shown in the last slide that was the example of a typical cell. So it actually comes in within a range. So it is another kind of cell. Here the concentration of sodium is some kind of different, whereas the concentration of potassium is some kind of different. Whereas in the last slide actually I have shown you a typical cell where the concentration was completely different from this case. Here the only perspective to show you this diagram just for the Na plus K plus or sodium potassium ATPase. So you have to just consider these nature or consider this representation that how the sodium potassium ATPase looks like. So there are basically two alpha units and there would be basically two beta units, which we will discuss in the next few slides. So we have already discussed that the sodium potassium enzyme is already having these two alpha subunits and two beta subunits, and as a whole it is named as alpha 2 beta 2 tetramer. And among these alpha and beta subunits, alpha subunit is the larger, which contains the ATP binding site, and one side of this alpha subunit the ATP binds, and at the other side there is the steroid inhibitor binding sites are present. And in the function of this pump, the alpha 2 unit is actually acting as a revolving door, which we will actually see in the when we will learn the detailed mechanism of this sodium potassium pump. Now you can see that I have shown you a typical X-ray structure of these alpha to beta 2 tetramer, and where clearly it indicates that there are two alpha subunits and two beta subunits. And actually see that this is the top view as per this X-ray structure. This is top view of this alpha to beta 2 tetramer, and actually this particularly filters the potassium ion to goes into this channel. And so this is not for your exam perspective. This is just for your knowledge, that how you should know that how the alpha to beta to tetramer looks like. Here in this diagram you can clearly see that there is particularly sodium binding side and there is particularly potassium binding side. So the sodium ions particularly binding at the outside at the inside of the cell, and then goes to the outside of the cell or the extracellular pod, whereas the potassium ions from outside of the cell actually it is binded and then comes inside the cell. And in the whole process the energy is coming through the hydrolysis of this ATP, where ATP is converted to these adenosine diphosphate and removal of one phosphate group. So this is a typical diagram of the subunit of sodium potassium pump, and the complete mechanism we will see later. Now we will go through the complete diagram or the complete mechanism of this sodium potassium pump. So in the function of this sodium potassium pump, first in the cycle, the transport of three sodium ions from inside of the cell to the outside of the cell and two potassium ions from outside of the cell to inside of the cell takes place. If you just look at this picture carefully, then you can easily note down that the sodium ions are just going outside of the cell, whereas the potassium ions are just coming inside of the cell. Because see that outside of the cell means that the outside extracellular part, and the other case is the inside part. In the first step, the sodium ions are going to bind with the alpha 2 units. How many sodium ions? In the first step, 3 sodium ions will bind to the alpha unit and then changes the local polarities of these alpha unit. Because see that when sodium is going to bind with this alpha unit, so it will be positively charged and then it becomes favorable to bind with a negative charged particle or element. Therefore, it is favorable to bind an ATP molecule, which is then hydrolyzed by ATPase, and this hydrolysis is catalyzed by magnesium 2 plus, and then 1 phosphate, and then it is converted into ADP with the removal of one phosphate group. Now, during these hydrolysis, the alpha 2 unit is phosphorylated. How it is becoming phosphorylated? Because during the hydrolysis of this ATP, it is converted to ADP and one phosphate is released or one phosphate group is released. And this alpha unit is actually binding with this phosphate group, that is HPO4 minus. Now this phosphorylation changes the conformation of the protein. How this conformation is going to be changed? Because this phosphate group is acting actually as a revolving door. You have just you do know how the revolving door works, because if you just push the door from one side, then it will revolve to the other side. So the phosphate group is just acting as a revolving door. And for this conformation change, presumably magnesium 2 plus plays an important role, because magnesium 2 plus is known to have a strong structure forming property. We have already told that there is some kind of structure conformational change is going on, and that process is actually called the eversion. And for this process, actually the phosphate group is very much responsible, because I have already told you that this is acting, or this is just trying to act as a revolving door. It is functioning as a revolving door. Now, in this new conformation, the sodium binding sites become open to outside, and the binding sites cannot bind sodium ions as strongly as before, because now it was open. First, the sodium ions, or the sodium binding sites were open to the outside, to the extracellular part. But when it is acting as a revolving door, now the sodium binding sites are open to the outside. From inside to outside, it is acting as a revolving door. Now when it comes to the outside of the cell, now the sodium ions, or now the binding sites cannot bind the sodium ions as strongly as before when it was inside the cell. So ultimately, this leads to the release of the sodium ions to the extracellular fluid. So the sodium ions is coming from the inside of the cell, and then it is released to the extracellular, or the outer side of the cell, or the outer part of the cell. Okay, so far we have seen that how the sodium ions are coming from inside of the cell to the extracellular part, just to maintain the concentration of this sodium. Because we have already come to know from the previous slides that the concentration of sodium in outside of the cell is comparatively higher than the inside of the cell. So therefore the sodium is coming from inside of the cell to the outside of the cell to the sodium potassium pump. Now the next step should be that the potassium ion should come from this outside of the cell to the inside of the cell. Why? Because as per we know that the concentration of this potassium ion inside of the cell is comparatively higher than it remains in the outside of the cell. So now the next step should be transfer of these potassium ions from the outside of the cell to the inside of the cell. So thus the open structure, now the structure is open because it just released the sodium plus ions. Now this open structure binds two potassium ion from outside of the cell, and this binding causes dephosphorylation. When it binds with the potassium ion, at the same time it is dephosphorylated, or it will release the phosphate group. Now from the it will release the phosphate group from the protein chain, that is the alpha unit. Now this dephosphorylation causes an eversion. Again an eversion will take place, that it will act again as a revolving door, and which will open the K plus binding sites open to the crystal, that is inside to the cell. And one thing I will try to mention here, that you have already know that 3 sodium ions are coming from inside of the cell to the outside or the extracellular part. Now why in the next step, 2 potassium ions are going to bind with this alpha unit. Why not 3 potassium ions? See that potassium ions are comparatively larger than the sodium ions. So as we consider the size, then in place of 3 sodium ions, 2 potassium ions can be attached. So therefore the transfer of these 3 sodium, in case of transfer of these 3 sodium ions from inside cell to the outer part, 2 potassium ions will be attached. Why? Because of the size. Because sodium's size of the sodium is comparatively smaller than the potassium. So where the sodium will be fitted, at that position 1 potassium ion cannot be fitted. So in place of these 3 sodium ions, only 2 potassium ions will be fitted when we will consider the size of the sodium and potassium ion. So therefore only 2 potassium ions will bind with that alpha unit, and when it will bind with that alpha unit, at the same time it will release the phosphate group, that is the dephosphorylation step. And when this dephosphorylation is actually occurring, again the eversion will be happened, and when the eversion will be happened, it will open the K plus binding side to the cistal, that is to the inside of the cell. Now the structure is open towards the inside of the cell because it undergoes the eversion for the second time when it acted as the revolving door. Now the K plus is not bound as strongly as before because the K plus attached with that alpha unit from outside of the cell, and then it acts as a revolving door and the eversion happened. After that, K plus is not bound as strongly as before, and these K plus ions are now released into the cistal, that is into the into the inside part of the cell. So as you can see that sodium goes outside the cell, whereas potassium comes inside the cell. And these things are happening at the cost of some energy. So from where this energy is coming? Through the ATP hydrolysis. And in that ATP hydrolysis part, magnesium 2 plus is playing an important role. And if you look at the concentration of sodium and potassium at inside and outside of the cell, then you will see that the sodium concentration inside the cell is much less, whereas outside the cell is much more. Whereas in contrast, in case of potassium, it is just the opposite. Inside the cell, the concentration of potassium is much more, and outside the cell, it is very less. So what is exactly happening here? That is, 3 sodium ions are coming from inside of the cell to the outside of the cell, and then 2 potassium ions are coming from outside part of the cell to the inside of the cell. So therefore 3 sodium in plus 2 potassium out will be converted into 3 sodium out plus 2 potassium in at the cost of some energy. From where the energy you are getting the energy? Due to the ATP hydrolysis. So the whole reaction would be 3 sodium in plus 2 potassium out plus Mg magnesium Mg ATP2 minus in presence of H2O will be converted into 3 Na plus out plus 2 potassium plus in plus Mg ADP minus, because the ATP will be converted into ADP with the release of one phosphate group, so plus HPO4 2 minus with additional H plus ion. Okay, so this is the complete reaction you will have to remember in the case of the sodium potassium pump. I have shown you once again a complete schematic diagram of the sodium potassium pump, and I will try to explain each and every step once again in this mechanistic part. So at the initial stage, as you can see that the pump is open to inside of the cell or towards the inside of the cell. Now, the step 1 is binding of sodium. That is, sodium binds with the alpha unit of the protein. So now it will undergo the phosphorylation in the step 2. That is, ATP hydrolysis is going on. From ATP it is converted to ADP, and that alpha subunit will be phosphorylated. as the alpha subunit will be phosphorylated, it will act as the revolving door and it will undergo in the step 3, that is the eversion. Now, through this eversion process, the conformation will be changed and the pump will be open towards the outside of the cell. Now, when the pumps are open towards the outside of the cell, the sodium ions are not bound strongly as before, and then it will release the sodium ions in step 4. Now in step 5, the potassium ions will try to bind with this alpha subunit. Now these two potassium ions will come to bind with this alpha subunit. This is the step 5 where the second half part of the cycle starts. When these two potassium ions bind with the alpha subunit, in the next step it will undergo dephosphorylation. It will release the phosphate group. Now when it will release the phosphate group, it will try to act as an revolving door once again. So in the next step, the eversion process will be happened. So through that eversion process, the pumps will be now opened towards the inside of the cell. Now when the pumps are open towards the inside of the cell, the potassium ions which will be bound, it will not bound strongly as before, and then the potassium ions will be released inside of the cell. Thus, the pump returns back to its initial state where it will again take 3 sodium ions again, and this catalytic cycle is going on. So this is the way how the sodium potassium pump works, and this is the detailed mechanism whatever I have tried to explain to you. Here I have shown you a schematic representation of the functioning of sodium potassium pump in terms of two different conformations, E1 and E2. E1 is taken on the basis of the pump is open towards the inside of the cell, and E2 is taken on the basis that the pump is open towards the outside of the cell. So now when the pump is open towards the inside of the cell, first 3 sodium ions will try to bind with that alpha subunit in presence of this ATP, that is ATP hydrolysis process is going on. ATP is converted to ADP through the magnesium 2 plus ion. Then in the second structure you can see that this E1 is attached with one phosphate group and 3 sodium plus ions. Now the eversion process will be happened. So after the eversion process, it will release 3 sodium ions. When it will release these 3 sodium ions, the conformational change will be happened through this eversion. So I have written here in the next step, E1 is converted to E2, and E2 is only linked with this phosphate group. So now when it will be converted into this E2 stage, it will then the potassium ions will try to bind with that alpha subunit. Now when the potassium ions will try to bind with that alpha subunit, it will undergo the dephosphorylation. So after this dephosphorylation, it will be converted into this fourth structure, that is E2, which is attached with 2 potassium ions. Now when it will be when it will come to the fourth stage, then again the eversion process will be happened, and after releasing these 2 potassium ions, it will come into the initial stage of the mechanism, that is E1. So here is the general representation of this functioning of sodium potassium pump in terms of these two different conformations, that is E1 and E2. I want to just remind you once again that I have taken E1 as the pump is open towards the inside of the cell, and E2 I have taken as the pump is open towards the outside of the cell. So I want to point out one thing here, that I have made one mistake, that is I have taken in the second structure, that is U1 is attached with OP3H. It should be OPO3H, because the functional group is HPO4 minus. In the third structure also, the OP3H should not be written there. It should be OPO3H. So this was a mistake I have done, and whenever you will try to write the mechanism in your copy, just remember this thing that you should replace this OP3H with OPO3H because the functional group would be HPO4 minus. Okay, so coming to the last part of the sodium potassium pump, I want to mention here that vanadate can interfere with the activity of the sodium potassium pump even at extremely low concentration, because at that concentration also it can inhibit the function of the sodium potassium pump. So the functioning of this sodium potassium pump can be malfunctioned with these presence of even a very low concentration of this vanadate. Why? Because these VO43 minus, VO43 minus, that is vanadate, and PO43 minus, that is phosphate moieties, are structurally similar, and this vanadate can compete with these PO43 minus. So in this class, we have learned everything about this sodium potassium pump. So what is basically the sodium potassium pump, how it operates, how the concentration is maintained, that is the concentration ratio of sodium with respect to outside versus inside, and the concentration ratio of potassium with respect to inside versus outside, how it is actually maintained through the sodium potassium pump, what type of enzyme is engaged with these, and how it operates means the complete mechanistic cycle we have already learned in this class. So thank you for watching this video, and if you have any doubt regarding this class, you can directly contact us through the website www.nsinst.com, or you can directly drop a mail at nsinst@gmail.com, or you can directly call, message, or WhatsApp us in the given number. Once again, thank you for watching. See you in the next class.